Meiosis is the cell division process that we will be studying this week. What is meiosis? It's a cell division process that will take place only in sexually reproducing organisms. The end product of meiosis is going to be the formation of gametes. Gametes is a general term for sex cells, such as sperm and egg cells. Now, why does this process have to occur? It has to occur to reduce the chromosome number in the gametes, making them haploid. Haploid, half the chromosome number of the original cell that produced them. One member of each homologous pair. This way, when the gametes unite during fertilization, the new cell that will be formed is going to be called the zygote. And the zygote will now restore the chromosome number of the species. For example, in humans, the gametes are going to have 23 chromosomes. So when the sperm and the egg unite, each one brings 23 chromosomes, one member of each homologous pair. And when they unite during fertilization, the new zygote will now have the magic number of 46, which is the number of chromosomes for the human species. Meiosis is also going to increase variation in the species, because during this process we will see recombination of genetic material that's going to occur during crossing over, also called synapsis. This will occur in prophase. Let's see exactly what we are talking about. Here we have a diagram of cells going through meiosis, the first phase of meiosis. As you can see, we use Roman numerals to identify the first phase, because this process is going to be a two-step cell division process. So here we see prophase 1. Just as in mitosis, the replication of the genetic material is going to occur in interphase. So in prophase 1, we already see the duplicated chromatids. And the difference being here is that they remain together. Here you can see a pair of duplicated homologous chromosomes forming the sister chromatids. And when they remain together like this, we're going to call this formation a tetrad. You only see tetrads in meiosis. When they are so close together this way, they can cross over and exchange genetic material, causing recombination. This only happens in meiosis, and it will exchange genetic material between the sister chromatids. In metaphase 1, the tetrads are going to line up at the center, at the equator of the cell, just like they did in mitosis. The difference being here now is, again, you have the tetrads. They are occupying the same spindle fiber. In mitosis, they were single-filed, but now in meiosis, you will have two pairs of chromatids on the same spindle fiber. These are the duplicated homologous chromosomes. They stay together. To be separated now in anaphase 1. This is when the homologous chromosomes will separate. Anaphase 1. Homologous chromosomes separate. In anaphase 1. So that when cytokinesis takes place in telophase 1, you will end up with two daughter cells that are now haploid, because you do not have homologous chromosomes in each cell. Each cell has one member of each of the original homologous chromosomes, and they are still duplicated. You can still see the sister chromatids held together by the centromere. This will go into, now these two cells will go into meiosis 2. Here is the second half of meiosis. Again, the spindle will form in the two cells that were the end result of meiosis 1. The spindle will form, and the sister chromatids now will align themselves single file, one member per each spindle fiber, just as they did in mitosis, but the difference being that these cells are haploid because you do not see homologous pairs in each of the cells. In anaphase 1, the sister chromatids will separate just like they did in mitosis, and then in telophase, this is anaphase 2. In telophase 2, the chromosomes now are going to be forming the nucleus as we had in mitosis. Cytokinesis will take place, and the end result is 4 haploid cells. Each one will be genetically different because of the recombination that took place during prophase 1 of this process. In this picture, you can see clearly what happens during that crossover. Here you have the tetrad, the duplicated homologous chromosomes. Here you can see them crossing over so that there is going to be exchange between sister chromatids. And it's clearly visible in this picture here. So that the resulting chromosomes, as you can see, no two are exactly alike. Each one has a different combination of genetic material. And this is a random occurrence, more likely to occur at the extremities, but each time that this process occurs, a different combination will be produced. The end result of meiosis, we mentioned before, is going to be the formation of the gametes. So we have 4 cells produced. In the male, we call this process spermatogenesis. And this is going to occur at puberty, as it will in the female. In the testes at birth, there will be cells called spermatogonial cells that will only divide at puberty during the production of testosterone. And those spermatogonial cells now will undergo meiosis, resulting in the formation of what we call a primary spermatocyte from one of the spermatogonial cells. This primary spermatocyte is diploid, and it will undergo the first half of meiosis, forming secondary spermatocytes that are now haploid. These will undergo the second part of meiosis and form spermatids. As you can see, 4 spermatids. Nothing is lost in the male during spermatogenesis. These will mature to form 4 sperm cells, each genetically different. In the female, the process is called oogenesis. There will be oogonial cells found in the ovary at the time of birth, and at puberty, they are going to undergo meiosis, 1 of them per month. Only 1 gamete is going to be formed per month as compared to the male, where they are forming 4 sperm cells each time the division takes place. On a daily basis, this is taking place. So in the female, we are going to form what we call a primary oocyte and a first polar body. That first polar body is going to disintegrate, and the primary oocyte is what will be released during ovulation in the menstrual cycle. The polar body is mainly there just to separate the genetic material. That primary oocyte is released, and the second part of meiosis does not take place unless a sperm fertilizes that primary oocyte. That will cause a second phase of meiosis to occur, and a second polar body will be formed. Again, these are going to disintegrate. That second cell that is formed now is called the secondary oocyte, and it will go on to form the egg cell. Again, the second division in the female does not occur unless fertilization takes place. So here you have the fertilization, the zygote, which is the new cell that's going to be formed due to the fusion of the egg and the sperm. And this zygote will now go on to divide by mitosis to form the new embryo.